if you have any questions later, yeah, please email me. And also, if anything comes up, hopefully you're all unmuted, and probably because there's not so many of us, if something doesn't make sense or you want to ask a question, please just do it orally. Or, you can always chat too, but it's probably easier for me to notice if you just say something. I think that'll be manageable. Okay. All right. So here's what here's an outline of what I'd like to go over. The length scales, the time scales, and the energy scales that are useful in biology. That is, how big are things? How long do they take to happen? How much energy do various things happen? And if you get some intuition for that, it probably helps to understand, interpret your own experiments, figure out what to do next, and that sort of thing. And so once we have those, and I'll spend a disproportionate amount of time on those 3 slides. And once that's kind of cemented in your intuition, I want to kind of reconsider, or just remind you, just so we're basically on the same page, the basic chemistry, and really basic chemistry that's almost ubiquitous in biology. Most of chemistry, most of what you learn in organic is kind of for specialists. But if you remember a couple of structures and remarkably few actual covalent bonds, it's enormously helpful for reading papers and that kind of thing. And I also want to give you a little reminder of the basic thermodynamics and kinetics that are kind of inescapably behind everything you look at in biology, even if you don't explicitly pay attention to that. And in particular, at least in the kind of a thermodynamic drive, what is everywhere in biological, at least in, even in dead biological things, is Brownian motion. It's hard to overemphasize how important thermal energy and Brownian motion are for biological things, even for things like mechanics. And so I'll dwell on that a little bit. And also, because this is a mechanobiology core, sort of boot camp, and you'll learn lots more about this. So I won't tell you very much about forces or that sort of thing. Anders will, and this will come up multiple times. But I want to show you a couple of examples of how big an effect forces, just mechanical forces, have on chemical reactions, even at sort of a single molecule level, because that's kind of been recently much more emphasized, and it's popping up as a useful thing everywhere. And then I want to kind of end, hopefully there's time to go over the mechanisms behind the non-covalent interactions. So most of the part that's fairly easy, the part that's fairly straightforward to learn in biochemistry and cell biology are the covalent interactions that hold stuff together, right? That's what, that's what sequencing's all about. That's what mass spec tells you, that kind of covalent assembly of atoms into molecules is kind of the basics of everything. But that is not what determines structure. What determines structure of almost everything in the cell are non-covalent interactions. And there are basically 3 or 4 forces of attractive interactions or repulsive interactions that are useful to keep in mind. And in the end, there probably won't be time, but I just leave you some cartoon pictures to remind you that almost everything that's a macromolecule in biology that makes up cell walls, the plasma membrane, lipids, everything, they're really just polymers of 4 different kinds of subunits. Lipids, polysaccharides, sort of sugar molecules and polysaccharides, nucleic acids and DNA and RNA, and amino acids and proteins. And I doubt that there'll be time at the very end, but maybe there will be. Okay, so let's launch into this part. So what one of the things that I think is really useful to keep in mind, and I say this because this has come up lots of times in things like oral exams of students who've been working on some cell type or some kind of organism or done some sort of reaction, and it becomes pretty imperative. They have no idea how big that thing is. And if you know how big something is, you can put really big, really important limits on what kinds of reactions you need to think about and what kinds of forces and interactions you don't need to think about. So here's one kind of useful scale taken out of some website. So the very smallest thing that is kind of a solid object is an atom. And atoms tend to be a few angstroms big. So angstrom-ish, so it's like 1/10 of a nanometer. It's a hydrogen atom, that's small stuff. Big things like buckyballs or things that are still covalent kind of monomer units, big molecules start to get to be nanometer scale. And I don't know why my mouse is failing, but I'm trying to see, sorry, I'm trying to see if I can, sorry, I can't.
Maybe your mouse is off screen.
Yeah, I thought it was, but it's not. It just isn't showing up. But so I may not be able to see chats. So if you have a question, please just do it orally. And then things like fairly big organic molecules like phospholipids on the left side, or lipid molecules. Those are a few nanometers big. Typical proteins and typical is hard to define, but kind of small compact proteins, things like albumin or hemoglobin or tubulin or something. Those things are maybe 5-ish nanometers big. So they're still less than you can see in a light microscope, but they're much, thousands of times bigger than the atomic kind of stuff that makes up organic reactions in the lab, for example. So those are sort of individual building blocks of cells. By the time you get to things that are arguably kind of livable, live or semi-live, very small things like viruses are 100 nanometers in diameter. And so 100 nanometers is at the lower end of what you can see with super resolution and a light microscope. So these things now are big enough that you can start to look at their structures by visualizing them. And so viruses are the smallest things. They're not all tiny. Some viruses are skinny and very long. So a virus can be a couple microns long if it's a filamentous virus, but things that are compact are kind of smallish. Bacteria are more or less typically a micron. And what's important is that I think this is important evolutionarily, individual bacteria are almost all about that size. There are no giant, blimp-like bacteria. What there are assemblies of bacteria that kind of fuse with each other to make very long objects that might kind of act coordinately. But the cell itself, the bacterium itself, is on the scale of a micron. And because mitochondria used to be bacteria, mitochondria are about the same size. So that's kind of it for the prokaryotic world. By the time you make eukaryotic cells, and even more plant cells, they get to be quite a lot bigger. So kind of very small animals or human cells like platelets or red blood cells, those have a diameter of a few microns. So definitely bigger than the bacterium, but not super big. And they don't have nuclei in them. So importantly, most nuclei, even though the size of the nuclei in different cell types varies, it's too bad a nucleus is not on here, but a eukaryotic cell nucleus is more or less 5 or 10-ish microns in diameter, just to pack all the DNA and all the polymerases and all the stuff that you need for replication. That nucleus is on the order of 5 or so microns, so about as big as a red cell itself, maybe smaller in a red cell, but not a lot smaller. So red cells don't have nuclei, at least human red cells don't. But then most big animal cells are bigger than that. So things like fibroblasts or endothelial cells, have diameters that are tens of microns. And plant cells could be much, much bigger than that. And some animal cells can also be much bigger than that. There are some stem cells that are 50 or more microns across. And so they have to be bigger than the nucleus that surrounds them. And some cells are way bigger than the nucleus that surrounds them. But these length scales are incredibly well preserved. And even the ratio of the size of a nucleus to the size of the cell, that's also conserved. So nature has gone to big lengths to figure out how to make things. And I think one of the main, or very big puzzle, at least in animal cell biology, and I guess this is true in plant cell biology, is that it is not clear what limits the size of a cell. How does an animal cell, how does a fibroblast know to grow until it's 20 microns in diameter, but not 100 or 1,000? How does a bacterium know to become a micron and not 2 microns? For the bacteria, it might be more similar. And similarly for the plant cell. And in diseases, there are lots of diseases where that size regulation goes wrong. There's all sorts of hypertrophies in cardiac disease. There are changes in the size of adipocytes during obesity or diabetes. And if you change the volume, the size of a cell, that has bad effects. And nature is very good at regulating sizes. But the principles of how the cell knows how big it is are not known. And then there are things that are on the highest end scale. And for some reason, because I can't use, sorry, let me just get out of here for a second. Oh, okay. I seem to have some, sorry.
So you're not able to advance?
I can't advance and I can't do anything. So I need to get.
Can you stop sharing for a second?
Yeah, try. Oh no!
There you go.
I can advance. I just can't. Yeah, I can't stop sharing because I can't move my mouse. So there's some kind of. Okay, so I'll try to go on, but unfortunately, I can't see the right end of the screen, and I can't see you. Okay, all right. So that's it for length scales. Now, time scales are also all over the place. And so this set of time scales is divided into two different scales of different sizes. So let's look at the top one across. So one of the, if you look on the left side, for things that have to do with cellular processes, stuff that goes on in a typical cell, some of the fastest kind of cellular events happen on a millisecond time scale. And by this, I mean not chemical reactions, but sort of whole programs of movement. So in one thing that we'll get back to in a second, but one of the consequences of bacteria being relatively small is that it only takes a couple of milliseconds for a protein, even a fairly big protein, to diffuse all the way across the diameter of a bacterium like E. coli. Smaller molecules like water, calcium, or sugars or something, they can diffuse across even faster, so in a millisecond or so. And one of the consequences of this, that probably has some truth to it, is that any chemical reaction where the reaction rate is slow enough, so moving of a reactant from one part of the cell to another can happen in a millisecond. For things like that, you don't need motors to move stuff around. Anything where diffusion is fast enough, you don't need directed motion. And so one of the arguments for why bacteria don't have myosin and microtubule motors and don't have directed motion in their cytoplasms is because they don't need it. And the reason they don't need it is because they're so small and diffusion is fast enough. And some things like the synthesis of big molecules also happen sort of on this same time scale. So one step during the polymerization of DNA takes on the order of 20, 30, maybe 50 milliseconds. So even just sticking one nucleotide onto the next in a growing DNA chain happens more slowly than it takes a nucleotide to diffuse all the way across the bacterium, right? Okay, so that's kind of the times. It's not always the time scale over which reactions happen. Some chemical reactions happen much faster than one step, but DNA polymerization is a complicated process, but that's about how fast it happens. And so that also tells you how long it would take you to polymerize a really long piece of DNA. It doesn't happen super fast because every step takes 20 or 30 milliseconds. If you have to polymerize 1,000 nucleotides, that takes a long time. Okay, so the other kinds of time scales are things like how long does it take something to be transported on a molecular motor. So a typical molecular motor in a eukaryotic cell, either dynein or kinesin on microtubules or most myosins on an actin filament, they will more or less move something up, about a micron in a second. Some are faster than others, some don't move a micron before they fall off the track, but generically, that's not a bad estimate. If you want to move a micron in a cell, it'll take about a second. And that movement becomes important now in transporting stuff in big cells like eukaryotic cells, because even though, like on the left side, it took a protein 10 milliseconds to diffuse across a bacterium, it'll take 10 seconds to diffuse across a fairly average-sized cell, like a HeLa cancer cell. So things that could happen fast enough by diffusion in a bacterium no longer happen fast enough in a eukaryotic cell. So protein diffusion across a cell is much, much slower. It's slower for two reasons. One is that the eukaryotic cell is big, but as we'll see later, it also takes a long time because the eukaryotic cell is also really viscous and crowded with stuff. Okay, so I'm going to, hopefully you can still see the slide. And I'm going to, yeah.
We can see the slide.
You can still see the slide? Okay. Mm-hmm. For some reason, I can't anymore.
Huh. Are you by chance on two screens?
No, I'm not.
Oh, okay.
But so I don't quite know what my issue is here. How has that happened? I'm sorry about that. Sorry, could I just stop and restart?
Yeah, maybe try to restart.
Okay. Okay. So let me try this again.
Okay, now we can see the screen playing.
Okay. All right, so I got to do one thing here. I'm going to minimize that. Okay. All right, you can see it again? Okay.
Yes.
All right, so that's, all right, so we've gotten as far as moving across an animal cell. It takes on the order of, 10 seconds or so. And then there's processes that happen, kind of metabolic things that happen in the cell. So molecules, when they're made, things like RNA and DNA, don't live forever. And the lifetime that a molecule in the cell lives is really important because it has a lot to do with memory. It has a lot to do with what happens during cell division. And there's no kind of single rule, but in a kind of vaguely generic way, big molecules that have relatively short lifetimes, by which I mean they're synthesized rapidly to do their job, and then they're degraded. So something like the messenger RNA in a bacterium that will, code for some protein. Typically, that message is made when the cell needs to make that protein, and when the cell has, started to translate that protein already and made it, it no longer needs the message to tell it to make more, and then it has to degrade the RNA. So in a bacterium, and more or less in most cells, a piece of DNA, once it appears, stays in that cell for something like 10 or 20 minutes, and then it gets eaten up. There are huge variations. Some messages have a much shorter lifetime. Some messages have a much longer lifetime, but they're not infinite. And proteins also have a finite lifetime. Some proteins are made and degraded relatively quickly, meaning in hours or so, and some proteins, once they're made, will live more or less forever. So, for example, collagen in some of your tissues, the collagen molecules you were born with, or maybe that's not quite right. But the collagen molecules that were secreted by cells to make your tendons when you were small are still there tens of years later. Whereas the collagen molecules in your mouth, if you cut your gum, the collagen molecules in your mouth live for a day or two. They're constantly made and degraded, but some collagen molecules stay around forever. So the cell is regulating that. But it's very expensive and it's very time consuming to make these macromolecules. So if you don't need to get rid of them, you don't get rid of them, but oftentimes you do need to get rid of them. All right, so that's it for the kind of the metabolic chemistry. And now the generation time, the turnover time of stuff. So live objects that divide fast, things like E. coli, they will replicate in about 20 minutes, half an hour, something like that. So those are prokaryotic cells. They will divide relatively quickly, so the generation time is fast. Some of the fastest eukaryotic cells, like yeast will divide not in 20 minutes, but let's say in maybe 80, 90 minutes, something on the order of an hour. So yeast will double much faster, but not infinitely fast. Right? So if it takes, so you can, the time scale that it takes for division is partly regulated by the control of the cell, because it wants to, it determines when to turn on the cell cycle and how fast to divide. But also it has this hard limit because it takes a certain amount of time to make stuff. So, for example, if it takes 20 seconds to make one nucleotide linkage in a piece of DNA or RNA, and you need to make very long polymers, you just have to wait for the replication machinery to make that piece of DNA. If you have to make a long protein, and some proteins have thousands of subunits, every amino acid step that's shoved onto the growing chain of a polypeptide, that takes a certain amount of time, and you just have to wait for the ribosome to crank those things out. And some ribosomes and some messages are translated faster than others, but still it takes a long time. And so that's why cell division is a fairly slow process. And things like big animal cells, they take, hours, tens of hours to replicate. Even a very fast dividing cell, like a HeLa cell, will take, 10,000, 100,000 seconds. So, close to a day to divide. Okay. All right. So that's it for the kind of the slow, big processes. And I don't want to spend too much time on the bottom part, but just to put this in some context, if you now think of the chemical reactions that underlie all this cellular stuff, those of course happen way, way faster. So some of the fastest things that happen in molecules, and these are of course really important for lots of biological processes, but things like bond vibrations and charge separation on a molecule, moving electrons from one oxygen to another, for example. Those things happen really fast. It's sort of femtosecond or picosecond time scales. And things like the reception of a photon in the photoreceptor of the eye, they have a big range of different steps in the photodetection cycle. So the photon first hitting the rhodopsin in the cell, that happens in a kind of nanosecond time scale. But what happens later to start sending the nerve impulse to your head to tell you that you saw a piece of light, that happens more slowly. And so that signal transduction time that tells you when the photon came out until your brain can interpret it, that takes close to a millisecond. And that reception time, all that signal transduction processing is what limits how fast people can see things. And different animals probably can see things faster or slower. And then there are things that are kind of really relevant to a lot of protein chemistry and a lot of cell biology, like right in the middle in this kind of microsecond time scale. A microsecond is about the time that a reasonable sized piece of alpha helix takes to transform into a random coil. Or if you're folding up a protein from a denatured random coil and you want to make it into an alpha helix, that takes something like a few microseconds, maybe less than a microsecond to 5 microseconds, but it's in that ballpark. So again, things like protein folding have a hard limit on them, that the thermal energy that's available for the macromolecule to explore its configuration space is limited, and that's about the amount of time that it takes to do it. There's not much you can do to make things faster. So as the result, on the right side, if you look on the top, the protein folding takes, milliseconds at the best, probably longer. So some proteins fold fast, some proteins fold slowly. It's not, I think, altogether clear how you predict which ones go which way, but they can take seconds to fold, or they can take milliseconds to fold. It depends on how complicated their structures are, how big they are, and things like that. And then things like ligand binding. How long does it take a growth factor to bind a growth factor receptor in a cell or something like that? That, again, takes milliseconds. So a lot of these cellular reactions don't happen super fast, but they happen, they have to happen they do happen on the time scale that the cell needs to respond. Okay. Any questions on the time scales? Okay. Okay. So let's look at
lots of head shakes.
Head shakes, yes or no?
No.
Okay. All right. Okay. All right. So energy scales. Energy scales also is really important. And if this isn't intuitive, then, and it sort of isn't, a lot of the stuff hasn't been intuitive in, at least for me, but it really helps if you get this, if you get some kind of hierarchy of how much energy goes into different things. So the kind of baseline energy that is surrounding anything that's biological in at ambient temperature is the thermal energy. So the thermal energy is about 0.6 kilocalories per mole, and we'll get into different ways of measuring energy in a second. But that's what KT is. It's 0.6 kilocalories per mole, or I guess RT, and that's about 10 times less than, or about 5 times less than a lot of non-covalent bonds. And towards the end, I'm going to spend quite a bit of time discussing the non-covalent bond options that molecules have. But so thermal energy is less than, but not an order of magnitude less than a single non-covalent bond between proteins or nucleic acids or sugars and stuff in biological conditions. So thermal energy is important on the time scale of non-covalent bonds. It's not important on the time scale of most covalent bonds. So even bonds that are um, well, sort of semi-high energy, like ATP hydrolysis, which is an important energy carrier. But the hydrolysis of the phosphodiester in ATP is nowhere near the strongest bond that there is. So it takes about 14 kilocalories per mole to hydrolyze ATP. It takes close to 100 kilocalories per mole to break a carbon-carbon bond. And that's also the, getting close to the energy input of a photon of green light, right? That's one of the reasons photosynthesis is so spectacularly good, is that the energy that comes from light is actually a lot. And then things like complete oxidation of some glucose, of one single glucose molecule gives you hundreds of kilocalories per mole. All right, so that's basically the kind of the energy scale to keep things in mind. And I'm not going to look at most of these things or go over most of these things with you because I assume you know them. But if you don't, or if there's something that's puzzling you, please email me or remind me later. But there's no sense in going over this too much. But just to very briefly remind you that you can understand almost everything about the covalent chemistry of most stuff in the cell just by knowing the different kinds of carbon-carbon, carbon-hydrogen, and carbon-oxygen bonds that exist. So if you remember what alcohols, aldehydes, carboxylic acids, and esters are, you're very far on the way to understanding the covalent chemistry of most of the biological macromolecules. Same with carbon-nitrogen bonds. I want to spend a little bit of time on phosphorus bonds because they're slightly less ubiquitous, but they're super important for not just ATP hydrolysis-derived energy, but as you'll probably see in lots of papers, they are kind of a central point in signal transduction. So, and also in the formation of the molecules in the cell membrane and that sort of thing. And there are really only two important phosphorus bonds. There are phosphorus-oxygen bonds and phosphorus-phosphorus bonds. The phosphorus-oxygen bonds are usually phosphomonoesters or phosphodiesters that are either part of the structural backbone of DNA, or they are ways to dangle a phosphate group from a hydroxyl group that's on the surface of a protein. So phosphorylation of proteins is one of the most common post-translational modifications, and maybe we'll see an example of that later. And the other thing is the phosphorus-phosphorus bond. So that's the phosphorus-phosphorus, or the phosphodiester bond in ATP, GTP, that kind of stuff, is the basis of this high-energy hydrolysis. And the only other really common covalent bond in biology are sulfhydryl bonds, either SH bonds or SS bonds. And these are more important for extracellular proteins than for intracellular proteins because, as I'll get to later, once you make a protein, for example, or a polysaccharide, or a lipid, once the chemical backbone of that object is made, that object is very rarely covalently attached to anything else. Almost all the interactions between a protein and something else are non-covalent, at least the stable ones. And the only exception are disulfide bonds. So in the extracellular environment, SH groups, which used to exist on the surface of intracellular proteins, once they're secreted into the oxidizing environment outside, they start forming sulfur SS bonds if they can find another SH bond to bind with, and the formation of these covalent sulfhydryl bonds is a big deal. Okay, not too much time to spend on that, but I leave this for you just to look at. And to kind of remind you of what you might see more in papers for sure is the various kind of nomenclature, some of which is not altogether accurate, but you often see phosphorus or phosphorylation of proteins denoted as some kind of little P in a circle. So phosphate itself, phosphoric acid, is just PO4 that's protonated to different extents depending on the pH. So the negatively charged oxygen on the phosphate is a very good nucleophile, and it can be very easily stuck on a hydroxyl group that dangles off of a protein or anything else. And so there are many, many enzymes that will take, not inorganic phosphate, but they will take the phosphodiesters that hang off of nucleotides. They will take that phosphate and stick it on to the dangling carboxyl group of a protein. And so that's usually denoted in the middle there. You take some kind of hydroxyl or an ester, or a carboxylic acid, and sorry. Look on the upper right. You take some kind of hydroxyl group and a phosphate group, and couple the phosphate through an oxygen ester bond to the carbon. Okay. All right. So let me just go on to the next thing. I want to remind you of a couple things of thermodynamics and kinetics. So, and this is mostly in the context of covalent chemistry. So there are only really two very important things to remember from whatever else you've learned from thermodynamics. And that is this equation at the bottom, that these two equations, one for thermodynamics, one for kinetics. If you just remember these, it'll be super useful. So the delta G, that is the change in free energy of some reaction, is equal to the sort of intrinsic free energy change for the two types of molecules, the product and the reactant being considered, and the ratio of the already existing concentration of products divided by the reactions. You've seen this a million times, and you may have forgotten about it. But if you haven't, take a look at that. Just take a look at it and make sure that the thing looks kind of normal to you, right? And so remember, the strategy is that a reaction will spontaneously happen if the delta G, the change in the free energy for something, is negative. And so there's nothing you can do about delta G0. That's just what's given you about the molecular structures of the products and the reactants. But what you can change, what you can change is the ratio of the products and the reactions. You can change the products and the reactions either by taking them away or putting them in, right? And so you want that logarithm term to be negative, right? So you want the ratio of the products to the reactants to be less than 1. So you can drive reactions by constantly taking the products away. And you'll see that again later, and that's a strategy that's involved in chemistry all the time. The other thing, which is uncoupled from thermodynamics, is chemical kinetics. So the rate at which something happens, that little k, is equal to some Arrhenius constant, some big A constant, something that that's just given to you by the nature of the chemical reaction being done, times this exponential term, e to the minus activation energy divided by kT. The activation energy is also something that's given to you by the molecular structure going on, presented in the reaction. So here in kind of cartoon form is what I tried to tell you about the delta G 0 plus RT times that logarithm of the ratio of products to reactants. Let's say that you have a reaction on the upper right in panel A Some kind of reaction between reactant X and product Y. And you let that thing go to equilibrium. So some X's will convert into Y's, some Y's will back convert into X's. And when you're left there, you have more X's than Y's, right? So the delta G is more negative going from Y to X than it is from X to Y, okay? All right, that's hopefully completely simple. But what nature uses all the time are strategies, usually based on ATP hydrolysis, where you now can take if you find a Y molecule, you can cause it to turn into Z, either by hydrolyzing it or by doing something else, right? So the nature has this arsenal of enzymes and all kinds of stuff to play around with what it can do to protein or to objects that it finds. And so if in a cell or anywhere else, there is a strategy to get rid of Ys whenever they appear, then you can make a reaction that starts off with a lot of X molecules and almost completely get rid of them, because every time you convert an X to a Y, you take the Y away and make it into a Z. And so this is a kind of ubiquitous strategy that you'll see in biology all the time. Okay, I hope that makes some sense. But again, it should be fairly near in your education and not. And I don't want to dwell too much on it. The other thing but what I do want to kind of hammer home is this thing which you probably have seen many times before, is that the rate at which something happens has nothing to do with the delta G. Right, the thermodynamic drive for something to happen, that is what happens at equilibrium, is not obviously coupled to how fast that reaction happens. Right, all kinds of things that are way out of equilibrium are very, very stable because there's no reaction pathway that will get the products to go to the reactants. So those reaction pathways are what are embedded into this Arrhenius rate constant. Okay, so read the bottom part. The rate at which something happens is, again, a function of the amount of the reactant you have at the beginning, but it's multiplied by this rate constant K. And K, again, is the Arrhenius law. That it's A, which is just a factor that depends on the molecular shape, and also things like the rate of collisions and how well the molecules are situated to bang into each other, which again depends on the diffusion rate and that kind of thing. And E sub A, that's the activation energy. So the activation energy is the amount of energy you need to put in to reactants in order to shove them together in the right configuration so that eventually they'll make the product. Even though the product is at lower energy, it wouldn't spontaneously necessarily get there. Okay, all right. So in kind of cartoon form, here's what this means. So let's say that you take a reactant that on the energy scale is a reactant Y, and it would spontaneously go to product X because product X has a lower activation, a lower energy, a lower free energy. So eventually it would get there. But in order to get there, the reactant has to overcome this activation energy barrier, which again has some complicated foundation in the way that the molecular orbitals are configured and the way that the reactants have to collide into each other in order to make the product. And often that's totally unknown. But what's interesting is that there are ways to change that. So there's no way to change the difference in energy between the reactant and the product because that's built into the structure of the molecules. There is a way to change the activation energy. And that's what's done by enzymes. So you can change the activation energy of a reaction and make it happen much faster by, for example, taking two reactants and putting them in some kind of device that slaps the reactive interfaces next to each other. There's a million ways to do this, but that's the whole nature of enzymes or any catalyst. It lowers that activation energy. Okay, so and what's important from a biological point of view, or from a kind of mechanobiological point of view, is that of the many things that can change that energy barrier, force is one of them. And this has been recently demonstrated in a bunch of contexts. And I'll show you one example of that in a little bit. Okay? Are we okay so far? All right. Okay. So how do you get over this energy barrier? The typical way you get over that energy barrier is thermal energy. So I told you that the thermal energy is 0.6 kilocalories per mole, but that's the average thermal energy of everything. And because of randomness, and this is Boltzmann's brilliant insight, not all molecules have the same energy. Some have way more energy than others. So if you look at a distribution, so a probability distribution of molecules with different energies, most molecules have about the same average energy. But there's this long, and some molecules have very, very little energy, but there's this long tail on this distribution. It's not a Gaussian-looking distribution at all. It has this long, stretched-out tail. And some molecules have way more energy than the average. And those molecules that have way more energy than the average, those are the ones that can climb up this energy barrier and do the reaction. And if you lower the energy barrier, you increase that kind of red area, the number of molecules under the curve that have sufficient energy to do that reaction. And the beautiful thing about the thermal heat bath and statistical mechanics is that if you get rid of the high energy molecules, that distribution doesn't change. It just redistributes. You start repopulating it with more high energy molecules. Doesn't happen immediately, but it'll happen. And so you just continuously put in molecules with sufficient energy to do a reaction. And so that's what enzymes do. They will take a reaction that might happen slowly. So for example, on the left is again a conversion of X to Y, which has a big negative delta G. So the reaction wants to go to the right side to make the blue molecules, but uncatalyzed, it'll do it very, very slowly. Every once in a while, as denoted by those arrows, every once in a while, a green one flips into a blue one, and a blue one flips into a green one. All that the enzyme catalysis does is make more of them be able to go one way or the other. It doesn't change the equilibrium. And it's the same if you look at the little energy diagram. You can off the product molecule, even though it's lowest energy, and it's in its own distribution of free energies, some of the molecules will have enough energy to bounce over that energy barrier and become a reactant again. Okay, so that's really all I wanted to tell you about that. But I wanted to dwell a little bit on where this free energy and diffusion, especially diffusion in water, where it came about and why it has such a dominance in biology. And largely it came about from a botanist named Robert Brown, who was studying pollen grains, and he noticed that the pollen grains in a microscope, if they were in water, they moved around. They didn't move around if you just put them on a glass slide, but if you put them in water, they moved around. And he didn't think that they were alive, like little animals. And he thought that they were, in my way of thinking, the phenomenon is the result of thermal molecular motion in the liquid environment. So this is an 1870. This actually wasn't Brown's idea. This was this other guy, Dassault's idea in 1877. he thought they weren't alive, they weren't moving themselves, they weren't providing energy, they were being kicked around. And it was really only until 1905, so not that long ago, that Einstein figured out how this worked. And the beautiful insight is, again, related to this energy scale. And so this is worth looking at for just a bit. I'm trying to see how much time we have. Yeah, this will take like 5 minutes to do at the most. Okay, so here's a unit of energy that's really useful to keep in mind, which is the unit of thermal energy is 0.6 kilocalories per mole, but that doesn't really help, at least often times. You can convert that thing to SI units. So a unit of thermal energy is 4 times 10 to the minus 21st joules. Okay, that doesn't help much either. But you can play around with the units, and on the kind of length scale of single molecules, because work is force times distance, or energy is force times distance, 1 unit of thermal energy is equal to 4.1 piconewtons times 1 nanometer. So I mentioned that, like, very small things, very small proteins are kind of nanometer sized, and 4 piconewtons, tiny force, but that's about the force that a single molecular motor can generate. So these are kind of useful, intuitive things to keep in mind. So if you remember that thermal energy is 4 piconewtons times 1 nanometer, that might come in handy sometime. And one of the things that it makes really clear is when thermal energy is important or Brownian motion is important and when it isn't. So for example, if you want, and I think we'll see some examples of this later, if you want to understand whether thermal energy is enough to really agitate something like a pollen grain, and remember, a pollen grain was a few microns big. And so that's a limit at where Brownian motion is important. And I think the reason Brown was interested in this is because if you put a big, dust particle or a piece of, carbon black or something in water, maybe carbon black you can see, but things that are sort of hundreds of microns big or a millimeter big, they don't move in water. It was this pollen grain that moved in water. And the explanation for why it moved in water is that if you look at the viscous drag of an object through a liquid, so the viscous drag of an object through a liquid is the viscosity times the square of the length times the velocity. So let's say we put it in water, that's about a centipoise of viscosity, and compare what the motion of a particle might be if it's 1 nanometer or 1 centimeter, right? So that's kind of 2 big limits. And let's imagine that the velocity of the particle is a micron per second. So that's about as fast as a motor can move things. Okay, so now if you just make a estimate of how much thermal energy is there available to the particle just from kT, and is that enough to overcome the viscous dissipation of a particle in water? Okay, so that's just kT divided by this viscosity times the volume times the velocity times the length squared. So that ratio is tiny for a 1 centimeter particle. So there's nowhere near enough thermal energy to move a 1 centimeter particle through water. That's why we never see, things like that jiggling. It's not enough to make something that's a millimeter jiggle through water, but it's way big enough. It's 1,000 if the particle is a nanometer. And it becomes, 1 if the particle is micron-ish. So that's why this pollen grain was able to dance around in the water. It's because thermal energy is enough to overcome the viscous drag. Okay, so that little estimate alone is enough to understand everything. And it's also about why Brownian motion works sometimes and not others. It's all a matter of what is the viscosity, what's the viscous drag in the medium, and how big is the particle. And this also translates into diffusion. So diffusion is also driven by thermal energy, and you can rationalize why things move fast enough in bacteria and not fast enough in eukaryotic cells again, by doing some simple calculations. I leave this one to you to take a look at. The last thing I want to show you is, I want to show you two things. One will take a minute to tell you, and the other one will take a little bit longer. Actually, will take no time at all. So not everything is driven by free energy of chemical reactions and thermal energy. A lot of stuff, a lot of chemical kinetics is now increasingly recognized to depend on forces. And one of the early really beautiful experiments of this is shown in the upper left here. That if you take an atomic force microscope or some kind of simple probe, and you try to pull apart two molecules or try to unfold a protein, the amount of time it takes to unfold a protein or to break the non-covalent bonds in the protein depends on how hard you pull on it. So if you don't pull on it at all, at least in this example on the left axis, it takes about 100 seconds for spontaneous thermally driven unfolding of this little protein domain. But the more force you put on it, the faster it unfolds. And that faster unfolding is enormously faster. So the y-axis up on the upper left is a logarithmic axis. So if you pull on something on this protein with 60 piconewtons of force, so that's about the force of a few tens of motors, you can unfold or break that protein bond not in 100 seconds, but in less than a hundredth of a second. So 4 orders of magnitude faster. So putting forces on molecules will have a big effect on the reaction time. But it's not always that the more force you put on it, the faster it unfolds or messes up. There are these recently discovered and really fascinating sets of bonds called catch bonds, which you can sort of understand a little bit in a cartoon form on the right side. So people have looked at this actin cross-linking protein alpha-actinin 4, which has multiple actin binding sites. So alpha-actinin 4 is that red and green dimerized thing in the middle, and it has these two, greenish binding sites, only one of which can contact the kind of tan-colored actin filament in the absence of force. It'll cross-link because 2 of those green balloons will stick to actin. But if you pull on the actin filament and the alpha-actinin is attached to the actin filament, you put force on the alpha-actinin molecule, and the tension that you put on the alpha-actinin molecule will move those 2 green balloons with respect to each other. And when that happens, the second balloon, which earlier was not able to bind to the actin, now suddenly does. So the more force you put on it, the stronger that bond is, because you've opened up more ways for the protein to bind to its ligand. And you can show that in a kind of a detailed way in other examples. In this case, it's the interaction between fibronectin, an extracellular matrix molecule, and integrin, its cell receptor. On the lower left, you look at the average lifetime as a function of force, and rather than monotonically decaying in those open circles you see in the middle, under the physiologic conditions where this bond actually forms, it's biphasic. Very small forces will cause this interaction to be more stable. And eventually, if you pull hard enough, you can destabilize it. So force has this very complicated and presumably biologically important role to play. Okay, I have one other thing to put into your intuition, which is that almost everything. So, yes.
Paul, I just wanted to give you a time check.
Yeah.
We have 8 minutes.
Okay, this is my very last thing, and it'll take like 30 seconds.
Okay, great. So
I just want to also to remind you that not everything is in dilute solution. Almost all these concepts that you learn in chemistry and biochemistry and thermo are for things that are in dilute solution, where the only thing that counts is the 2 molecules that are reacting with each other. In cells and most biological things, that's very much untrue. So here is a kind of a colored picture that's accurate to scale of how crowded a cell is. And so cells are not bags of water in which there are chemical reactions. Cells are just crowded with all kinds of stuff in them. And the cytoskeleton, organelles, all sorts of molecules. And so the consequence of that is that these kinds of diffusive motions and this sort of moving around, they are constantly being interrupted by the molecules bumping into each other. Okay, so I leave it there, and I think the end of the slides is a couple of examples of things that you might want to remind yourselves of, but that we don't need to go over now. Okay, so I'll stop there and stop sharing my screen. Okay. All right.
And I think we have a couple minutes for questions if anyone wants to pose a question.
Okay, so one question I have is that we usually hear about how enzymes decrease the activation energy and thereby make reactions easier, but do they do the opposite as well? Do they increase the activation energy and thereby make it harder to?
Oh, that's a good question. So I don't know whether there are ever, I've never heard of a, it's a really interesting question. I don't think I've ever heard of a, oh no, there are things like that. I don't know that in animal cells there are things that will increase the activation energy. You can sort of imagine doing that. Inhibitors will do it. You can prevent something from being degraded by binding to the site that the enzyme or the degradation machinery will try to get rid of it. Yeah, so there's that. And then there are these things which I think plants have, but there are antifreeze molecules that will prevent animals from freezing, even though at equilibrium they're cold enough that they should freeze. And they will do that by every time an ice crystal forms, they will stick on the surface of the ice crystal and just prevent it from growing. They don't change the equilibrium for freezing, but they just make the freezing process infinitely slow by messing it up every time it tries to crystallize. But other than things that will block a reactive site, I don't know of a mechanism that will slow down a reaction. I mean, that's a great question. I'm not sure that I've... Anders, have you ever heard of anything? I don't think I have. It's kind of interesting. No, that's a really good question.
Okay.
Anders is here, and Anders, you're muted. It looks like he was trying to answer.
Okay. Hi, Paul. I heard your part of your answer, but not the question.
The question is whether there are any enzyme-like things that increase the activation energy of a reaction rather than decrease it.
Wow, I've never heard of that, but it could be advantageous because a lot of times cells work by negative regulation. So why not use enzymes to help that? Neg enzymes.
Yeah.
Mm-hmm.
Can I ask a question about Brownian motion?
Oh, yeah, please.
So you talk about how the thermal energy dominates the viscous energy, but I've also learned a lot about like sperm, for example, and flagellum when they go through low Reynolds number flow, that the viscous effects dominate. So the viscous is a lot higher. So is that still dominated by the thermal energy, even though it's so high? And is that, like, how do those scales relate, I guess, is what I'm asking.
Yeah, so at least in any sort of flagellar-driven motion, that's all using ATP hydrolysis as the energy source. That thermal energy would no longer be sufficient to move a sperm cell through anything that's viscous enough. And so you have to burn chemical energy, and you have to sort of get out of equilibrium in order to do that. And yeah, so I think that, that's an important distinction. So low Reynolds number, presumably always, thermal energy is not going to help you.
So I think that if there aren't more questions, I'll give that a second, but we might allow the students 2 minutes to stretch their legs.
Sure. Yep.
Anders is here, ready for his next, for the next lecture on measuring forces.
So patients, why don't you just give me the go-ahead when we're ready to go, and I'll start it up.
Okay, and Paul, would you be willing to answer questions if students, by email, if students have follow-up questions?
Oh yeah, absolutely, sure, yeah, yep.
Okay, well, thank you very much for your time this morning.
Great, okay, all right, okay, thanks, bye-bye.
Thanks, everyone.